Hello, everyone. I'm Alberto Fierro, the Consul General of Mexico in Boston. Welcome to one more of our conversations with Mexicans in New England, where we want to portray how Mexican academics, scientists, artists are contributing to the knowledge and cultural life here in New England. Today, as you know, our special theme is Advances on the Study of Parkinson's Disease. And we have a special guest, Dr. Luis Fonseca Ornelas. Luis, thank you so much for accepting to be part of this conversation.
Well, hi everybody, and thank you all for having me. It's an absolute pleasure and an absolute honor to be invited to be here.
Dr. Luis Fonseca Ornelas is Research Fellow in Neurology at the Harvard Medical School and Brigham and Women's Hospital. He completed his PhD in structural biology at the Max Planck Institute for Biophysical Chemistry at the Göttingen University in Germany. He completed his Bachelor of Science in Biology at UNAM. As part of his research in structural biology, Luis studies neurodegenerative processes in diseases such as Alzheimer's and Parkinson's. Luis, can you tell us about your background? Why did you come to Boston if you were in Germany? We know your education was first as a biologist. How was it that you became interested in Parkinson so much as to devote your career to studying it?
Well, first of all, I think my love for science comes from my early years in middle high school. You know, I had very good teachers in biology, and very soon it was clear to me that my favorite subject had to do with not only biology but neurobiology in particular, right? How did our brains work? How our brain makes what we are, and very specifically, what I liked early on in my career was how do the cells in our brain die under certain conditions. And of course, one such condition was Parkinson's disease and Alzheimer's disease. So I was always very interested in that. So when I went to, after being in Germany, I was lucky enough to be in a very good research institute there, and I wanted to continue with my career. And of course, there are very few places that combine, these. I mean, what Boston and Cambridge have is this: they have a lot of very good scientists in a very small area, right? So it's very easy to, if you have an idea, it's very easy to just go to your neighbor and tell them about this idea, and chances are that they're going to know somebody who can help you, like develop this idea into something more, more tangible, more that you can actually work with. So I truly believe that there is no place like Boston and Cambridge to do, like basic scientific research, right? Just the amount of people, the amount of resources, in terms not only about money but about people. I think there's no way to compete with that, and of course I wanted to try that and come here, and yes, that's how I did it.
And did you come first as to do a postdoc, or you landed a job before coming?
No, so my idea was, when I finished my PhD, I was looking for jobs all over the place, and I interviewed here with Dennis Selko, who's one of the most well-known researchers and clinicians in Parkinson's and Alzheimer's disease, and I spoke with him, and he was very, he's very forward, and yeah, he invited me to come, and he has been super supportive of me in my career. So yes, that's why I came here.
Fantastic. With us is also Paulina Ramirez, who is in charge of the innovation area at the consulate. Paulina, do you want to?
Yes, of course, Consul. Thank you so much. And hi, Luis. Thank you for joining us today. So I'm sure that most of the people here have heard about Parkinson's disease, but could you tell us what it is? We do know that this disease happens when the neurons that are located in certain part of the brain that control our movement, they cease to function or die. But we do not know why do this. Why does it die? What are the most probable causes for this area of the brain dying?
Well, that's a fantastic question, and that's a question that even though many people have, like poured themselves trying to answer it, I mean, we don't know. We know that there is a very small population of cells in the brain called the dopaminergic neurons in the substantia nigra that die, and when they die, people develop symptoms of Parkinson's disease, right? And these symptoms are very well known, right? I mean, the cardinal symptoms of the disease are these resting tremor, right? This difficulty with moving and like these fine movements. These are the cardinal symptoms that are very, like visual, that are very easy to spot. And I'm sure people have seen Michael J. Fox, for example, right? He has Parkinson's disease and he has this tremor. I'm also sure that people remember Muhammad Ali in his later years, right? He also had this tremor, this very evident tremor. But as I said, like, yes, it's very true, like cells in the brain die, but we don't know the cause for that. Not yet, at least. I mean, we are trying to, that's what many groups around the world are trying to unravel.
And what are the probable causes science has been able to find?
Right. Well, as I said, yeah, it's really hard to pinpoint it to something, right? So other than a few cases in which, for example, people that have been exposed to certain kinds of pesticides that develop, like these Parkinson's disease-like symptoms, it has been really complicated to pinpoint a single cause for this, and I think that's what makes it so hard to study, right? Because we don't even know for sure how this happens.
And what are some of the symptoms? At what age does the disease start to develop? Is there a certain age? Do we know already if it is hereditary or not?
Right. So there are two types of Parkinson's disease, right? With the one type, which is the most abundant, the most common one, it's called idiopathic Parkinson's disease, and there is no clear hereditary component to this, right? It just happens to people that are, like above 60, 65 years of age, and like many other diseases like Alzheimer's, of course, but also like disorders in the heart. Like the older we get, the more chances we have to develop any of these diseases. So I would say that, yeah, around 65 years of age is a pretty common initiation of this disease. And as I said, like, even though most people have what it's called this idiopathic PD, a small percentage of people do have what is called a familial form of Parkinson's disease, in which a mutation in certain genes predisposes a person to develop Parkinson's disease, right? It's a hereditary form, and most of the times, these forms are actually much more aggressive than the idiopathic form. So if you have any one of these mutations, the chances of developing Parkinson's disease are much higher. But not only that, it's not going to happen when you're 65 or 70. It's probably going to happen, there are some cases in which it happens when they're 25, 30 years old. Again, right? For example, Michael J. Fox, he has a mutation in one gene, and he developed Parkinson's disease at around 35 years of age. right? So it's much more aggressive when that happens.
And how can you detect it at an early stage? And
well, that's actually a fantastic question, because another, yet again, this is one of the areas in which most people are trying to develop. It's called a biomarker, right? We need to find a way in which, you go to the doctor and you get some blood drawn and to detect the disease. But so far that has been mostly unsuccessful, and that is a huge concern, because these cells that I was telling you about, these neurons that live in the substantia nigra of the brain, which is a small region in the, very deep in the brain. When a person goes to the doctor because they feel like they have this resting tremor, or they feel like they have lost the smell, for example, that's also very common, right? That they can no longer smell. When people go to the doctor with these symptoms, around 85% of the cells living there in this part of the brain are already dead, right? So it's really late in the development of this disease that we even start to see the symptoms, right? So it would be absolutely essential to have some kind of biomarker, right? Well before that, so we could, not until it's too late, but like before the symptoms even appear, that we could start to treat people. But that, as I said, that is one of the main areas of concern in Parkinson's research.
when I studied biology 100 years ago, I do remember hearing or learning that every day we lose a lot of neurons every day. But in this case, it's some particular type that die.
That's correct. Yeah.
Movement.
Yeah, that's correct. So the substantia nigra neurons are also like dopaminergic neurons because they make dopamine. And the problem is not necessarily that they die there. The problem is that they no longer release dopamine into another region of the brain that is called the striatum. And this part in the striatum is the one that, among many other things, it's a very complex part of the brain, but among many other things, it controls fine movements, right? So if you no longer had the dopaminergic input from the neurons because they died, then you can no longer control fine movements, right? So that's why this happens. And also, like that's one of the issues, right? Like, sure, you may lose some brain cells in some parts, but when you lose 85% of cells in one particular region, that's when the real trouble begins for Parkinson's disease.
Paulina?
So why does the accumulation or lack of this dopamine become so dangerous to the brain? Is Alzheimer's disease similar to the Parkinson's?
Right. So as we said before, right? So it's not very clear how like the early stages of these diseases. Why do people develop these diseases? But one thing that seems to be clear is that there is in these particular neurons, there is an accumulation of one protein that is called alpha-synuclein, right? And that's exactly what I studied even back in Mexico when I started my career in Mexico and in Germany, and now here in the States. I studied this protein that is called alpha-synuclein. And when alpha-synuclein stops doing what it's supposed to be doing, it starts to clump, and it starts to clump in many neurons of the brain. But these particular neurons that control the fine movements, that are dopaminergic, are particularly sensitive to these accumulations. So these proteins start to accumulate, and then the cells start to die. And that's precisely one of the key targets for like all of the research trying to cope with this disease, and many pharmaceutical companies. How can we prevent this protein, this alpha-synuclein protein, from clumping together and killing the cells? So that has been one of the main concerns. And Incidentally, and as you very nicely said, Alzheimer's has a very similar process, right? Also, we don't know how or why Alzheimer's happens, right? But in Alzheimer's, another protein called A beta also clumps and accumulates, right? And the neurons die. And in Huntington's disease, another neurodegenerative disease, Huntingtin, another protein, also accumulates in clumps together. You know, ALS, amyotrophic lateral sclerosis, the Lou Gehrig disease, also neurodegenerative disease, has an accumulation of another protein called TDP-43, right? So accumulation of proteins and this clumping of proteins seems to be a common mechanism to which particular sets of neurons are sensitive to, right? So, so that's why, as I said, our group and many groups studying these neurodegenerative diseases are trying to first understand, because the truth is that there's still a lot to understand, and we don't know how these mechanisms work. And once we have a bit more information about how these mechanisms work, then try to, you know, do something to prevent that.
Is it true that caffeine consumption reduces the risk of suffering from Parkinson's disease?
Well, it's very interesting because there is a correlation. There is a correlation.
Drinking coffee like 3 weeks ago, so I'm back to it.
You should probably go back to no. So first of all, there is a correlation between people who drink more coffee seem to have less propensity to develop Parkinson's disease, right? Now, it is very important to before you go ahead and drink another 4 cups of coffee, it's very important to know that this is a correlation, right? And correlation does not imply causation, right? So the fact that we have this correlation doesn't imply that we should absolutely go and drink it. Now, if you already drink, you know, like, I mean, tea also has caffeine, right? So, and tea and coffee are, for the most part, healthy things. So, you know, drinking a couple of cups of coffee or tea per day may, you know, like prevent you from getting Parkinson's, but, you know, like, it's certainly not going to hurt you. So maybe 1 or 2 cups of coffee a day, or tea. Maybe tea is better.
Okay.
But yeah, that's good. You
made us all coffee lovers so much more happy again right now. So does this
drinking because I was taking too much.
Sorry, so, I couldn't hear you.
That I stopped drinking because I was shaking too much. So I wanted to know if the coffee had to do with my shaking, or if I have to go to Parkinson's disease.
Well, one way that you could, you know, again, correlate it is, you know, that many patients of Parkinson's disease say that the first thing that they noticed, it's not even the shaking of the hands, but the loss of smell. Yeah, so that's one of the first symptoms. And then another one is, you know, like this shaking, most of the time starts in only one side of your body, right? So it's only the right hand or only the left hand. And then as the disease progresses through many years, then it goes to the other side of the body. But maybe it's just that you were taking too much coffee. So
hopefully, yes. Paulina, you were going to ask something?
Oh, yes. So it's also been said that diabetes has shown an increase of the risk of suffering neurodegenerative conditions like Parkinson's.
Right.
What's your take on this? Is this true?
Well, again, right, just like with coffee, I mean, with caffeine and Parkinson's, people see this correlation, but the mechanism by which caffeine could, you know, like help or prevent Parkinson's is a lot less clear, right? So it's only a correlation. With diabetes, it happens more or less the same thing, right? So people who have diabetes have developed Parkinson's or other neurodegenerative diseases more often than people who don't have diabetes, right? So these could be just a matter of chance, just a correlation that has no mechanisms connecting the two. But the truth is that one of the main issue with diabetes is that people have more glucose in their blood. Doesn't matter if it's diabetes 1 or diabetes 2, right? We just have more blood, more sugar in our blood. And the blood vessels that feed the brain are very sensitive, are very, very thin, very sensitive, and an accumulation of sugar over time tends to be toxic, right? And so it is entirely possible that after many years of having higher levels of glucose in the blood, the brain will suffer first. So yes.
Wow, that's interesting. So no excesses, too much coffee is bad, and also too much sugar.
Yes, no, too much coffee is okay, not too much, but too much sugar, definitely a bad thing, definitely.
Terrible. Wow.
And really so you tell us more of your specific research and how do you do it? I remember a couple months ago before COVID, I did go to this brain center at MIT, and I was impressed even with the artwork of this brain that you must have seen there. But your research is done in a hospital. How do you do it? Are you seeing patients? How is it that a scientist like you does this type of research, and what do you focus on particularly?
Right. So I'm not a clinician, so I'm very lucky that I'm in a hospital, because being there in a hospital, you have access to actual, patient-derived tissue, right? So something that is very precious, right? People that have gone through this disease and they take a very valuable decision that when their lives end, they donate their brain for research, right? And we have had access to those brains, right? Which is a precious thing, because it's the source of everything that we want to, as I said, first understand, and ideally, eventually try to stop. So what I do is that I take the brains of patients that have had this disease, or Alzheimer's, or some other ones, and then I study, I separate the brains into very specific components of the neurons, but also I chop the neurons into its most essential components, and then I look at when two neurons talk to each other. It's called the synapse, and so I look at the synapses of the brains of these patients, and within those synapses, within this connection between two brain cells, I look at the shape of this protein that I was talking about before, alpha-synuclein, that is involved in Parkinson's disease. And I look at the shape with the help of a technique that is called nuclear magnetic resonance. So I take these cells and I put them inside a very powerful magnet that allows me to see the shape, the structure of this protein alpha-synuclein at atomic resolution level, right? So I can see very intricate parts of this protein in this very complex environment of the synapse, right? Of the neurons talking to each other. And what I'm trying to find is how different and exactly which differences are between the alpha-synuclein that is in a normal brain right? A person that didn't have Parkinson's disease, and the alpha-synuclein that is inside of a person who had Parkinson's disease, right? And what I hope is that by finding differences in the structure of alpha-synuclein in disease and in health, we can, more accurately design compounds that prevent synuclein from going to this proper way of being into another toxic way of shaping. So that's pretty much what I do. And as I said, like many of the things that I do, many things there at the hospital. I do many others at the at Harvard, because they have instrumentation that is also fantastic. And I'm also very lucky that I'm right now doing another type of experiments at the MIT. And so, as I said, these type of resources, this type of availability is absolutely fantastic here in between Cambridge and Boston. So yes.
Wow. And how much, once you have access to one of these brains, how much time do you have to work on it?
Well, it depends on the particular thing that I'm doing. If it was already preserved, then it doesn't matter, because it's normally preserved at either -20°C. So it's like ice. So it doesn't really, as long as you keep it like that, it's fine. But for many of the things that I used, I need it to be very fresh. And so what we do in those cases is that the Mass General Hospital, I also collaborate with Mass General Hospital, and they have this very large cohort of patients that are enrolled in this, like this whole process of donating their brains. And so as soon as the person dies, we get a call, and then we go with the medical specialist who's performing the autopsy. And so as soon as they perform the autopsy, they take the brain out, they cut the pieces that we need to study, and then I take it back to the hospital, and I start to do my thing. So between the time of that and the time that I'm starting my experiments, there can be anything from 4 to 48 hours. So it's, again, something absolutely invaluable that we have access to here.
Wow.
We have a question from the audience, so I'm going to read it right now. It's from Felix Gomez, and he's asking, the CRISPR technique developed in 2020, developed by the 2020 Nobel Chemistry Prize winners, and which works with genome editing, could it be helpful in the treatment of Parkinson's? What's your thoughts on that?
Well, it already is helpful. That technique is, well, it, as Felix said, it won the Nobel Prize a couple of weeks ago, because it allows researchers to very specifically change the DNA sequence of a particular organism, right? So that allows you to perform experiments in which you change something to see what are the effects that these have. So CRISPR techniques are used all the time in labs, not only in neurodegeneration research, but also in many areas. What we do in particular with CRISPR, for example, is that we take the skin cells from a patient that has Parkinson's disease or Alzheimer's or whatever, and then we culture these cells in the lab, and then we turn them into neurons, right? And so they look like neurons, and they are from this patient, and they have all the information that the patient has in their brain. And so what we do is that we take CRISPR, and then we change a small little thing in the DNA of these cells to see if we can either exacerbate the effect that we see in these people, or if we can somehow like fix it. And so yes, everybody uses CRISPR. yeah, it's a super well-deserved Nobel Prize that it's already changing the way that we do research.
Right now, there is no cure nor medication that can reverse the effects of Parkinson's disease. But what are the treatments available? Do you believe there will ever be a cure for this kind of neurodegenerative conditions?
Yes, I absolutely believe that there will be a cure for Parkinson's disease and for Alzheimer's disease, because there's a huge amount of very well done research that has no other objective other than cure these diseases, and I'm sure that will happen. I don't know if it'll happen within my lifetime. That's another question, but I'm certain that we will be able to overcome these disease. Now, what was the other question, sir?
Treatment.
The treatment so far. Yes, sure. Well, the treatment so far, there are two types of treatments. One is that, as I said before, the dopaminergic neurons in the brain die, right? And so we no longer have dopamine where we are supposed to have it. So what people do is that they put dopamine-like molecule in a pill, and then you take it, right? It's called L-Dopa. And so yeah, people, it's replenishment way of dealing with this disease, right? Another one is that it's called deep brain stimulation. And so what they do is that they drill two little holes in your skull, and then they put wires all the way down to the region where the dopaminergic neurons are already dead. And these little wires are connected to a little battery that every X amount of time releases an electric charge and then promotes the activation of those neurons. And that is absolutely fantastic, because that is, for many people, it's a game changer in terms of dealing with the disease. However, as good as these two treatments are, they deal with symptoms. They don't cure the disease, right? So of course it's naturally better than nothing, of course, but there's still plenty of things that we have to do to reach a cure for this terrible disease.
So these treatments would only delay the advance of the disease, or
no, not even that.
Oh.
They don't delay the disease. Keeps on advancing, and what happens is that people start taking, say, one pill of this L-Dopa per day, and then by the third year they have to take 10 pills per day to more or less see the same reduction in symptoms, right? So these therapies do not solve the issue. These therapies just just treat a bit of the symptoms, and they, of course, they improve the quality of life of the patients, but they are still not cured.
Wow, but it's interesting and wonderful to know that there is hope that that kind of a medical condition can be someday cured. Now, tell us final words. You mentioned before we started the program how happy you are in Boston. I don't know. I've never lived in Germany, so I don't have that experience. But you've been here for 3 years and a half. We lost the direct flight to Mexico, unfortunately. We hope to have it again maybe next year. But tell us a little bit more about your life here, or how do you see your future in this research and this fantastic innovation hub?
Well, I mean, I have been really happy here in the States, because there, like, when I moved to Germany, it was such a change from Mexico to Germany, right? In terms of the food, in terms of the weather, in terms of the culture, in terms of you know, like how things are done. It was just so different to me. And so when I, you know, coming here to the States, I feel like, you know, in many ways, and that may be because, you know, like Mexico is under such a huge influence from the States, but I felt like you seem more like Mexico, right? You could find more Mexican places to eat. That's really hard to do in Germany. Yeah, I really have really enjoyed my time here, and while, you know, like, of course, I as I said before, right? So if somebody wants to do some, you know, some kind of scientific research, I'm sure there is no better place to do that. At least in the, you know, like I'm familiar with biomedical research, right? So I'm not sure about any other, you know, like fields, but about biomedical research, there are few places in the world as good, as competitive as Boston is. But again, right, as much as I like that, and as much as I enjoy doing research, I am looking forward to eventually go back to Mexico and, you know, be able to, like, I mean, that's where I was born, and that's where I, you know, like, everything that I know is. And so, of course, eventually I would love to go back there and, yeah, start my own lab there and whatever. So let's see what happens.
Yeah, hopefully. Paulina, do you have any more questions?
I don't have any more questions, but I found this conversation just fascinating, and I've learned so much. Thank you so much, Luis.
Oh, thank you.
Thank you, Luis, and hopefully we'll get to meet each other soon once the pandemic is over, and we'll have something here at the residence that you might have passed through many times.
I have been there a few times. Yes. Okay. Well, thank you so much,
everyone, for joining us, and continue joining our conversations with Mexicans in New England every Tuesday at 4 o'clock. Thank you.